So yeah. Thank you so much, sir, for joining. So friends, this is probably 86th Sciversation, if I count it correct. And thank you so much. And this is a very special Sciversation with Dr. Moinakl Banerjee. So sir needs no mention. I had a good fortune in interacting with him on many occasions. Just a very brief introduction into his illustrious career. He started his undergrad and graduate education from MLSU in Udaipur, and then he worked for All India Institute of Medical Sciences and CCMB. So he was my senior many ways, almost 12, 13 years senior. And then since almost 30 years, he has been at RGCB, right from his heydays of Scientist C to Scientist G until his retirement this year. And it has always been a kind of a great honor reading his publications and amazing scientific journey that he has guzzled through over the last 3.5 decades. One of the highly cited articles of his is through genetic sweeps and genetic gateways to COVID-19 infection, which appeared in the FaceUp journal, which we read it with great interest in mid 2020 when the COVID-19 happened. And apart from that, he has always been working on genetic variation associated with neuronal dysfunction and neurobiology related diseases. And apart from that, he has always worked towards myriad of subpopulation genetics, including the epigenetics of schizophrenia and other related diseases largely associated with South Indian subpopulation. So with this little introduction, thank you so much, sir. And he has mentored countless number of PhD fellows, where his lab largely works on genetic variation, not with respect to humans, but there was also some kind of studies that he has worked on the genetic variation that is sitting in lions and tigers with Lalji Singh. Yeah. Thank you so much, sir, for your kind time and energy today. We'll start with a quick presentation on your career for the first 10 minutes, and then we'll have some Q&A session extempore from my side, followed by the audience. Over to you, sir, please.
Thank you, Prashant, and thank you, this Bioclues, for organizing this event. And also, I think it's Bioclues is a movement which has been spearheading the activities of popularizing science. And I thank the entire community for giving me this opportunity to interact with you. So what I'll do is I'll orient you about a brief of my background, which Prashant has already mentioned it, but I'll still share my slide. Uh, can you see?
Yes, sir. Yes, sir. Please. Yeah, you can make it full screen, sir. Yeah.
Yeah. Yeah, can you see me?
Yes, but not full screen. Yeah. If you can make it full screen. If you're using MacBook, there is a glitch with that. You cannot make it full screen until you re-log in again.
No,
But the slides are...
I'm using a Windows system.
Can you move to the next slide, sir? We can see your slides. Yeah. Yes, please. Yeah, please go ahead, sir. Please, yeah.
You can see it, right?
Yes, sir. Yes, please.
Okay. So, Is it clear now?
No, sir. I think it went it went differently. Your slides have become smaller. I don't know why. Is it also for others or is it only for me?
Wait. Then,
For sir, sir, can you just press F5 from your keyboard? F5.
Yeah, there is also a glitch with Firefox, Santosh. That is what probably is the problem. Yeah, I don't know. Yeah, F5 would work. Yes, yeah. Again, it is coming in the previous way.
huh?
Yeah, yeah. You can probably start, so that should not be a problem. Please go ahead. Yeah,
Yeah, but I can't see any one of you there. That's the problem there.
yeah. That happens when you're in full screen. Yeah, but usually, yeah. Can you see two windows if you're using Chrome? You can click the other window.
Yeah. So
Then you have to make. Yeah, yeah. It's okay, sir.
you
It's
can
okay.
see the
You can
slide.
go ahead. Yeah, you can go to the next slide and start. Yeah, please.
So yeah. So my journey to genetics or whatever I do was very complex. My background was not classical genetics. I started working with first of my mentor in the MSc, who actually taught me about the taxonomy. Then from MSc, I went on to do my PhD in Udaipur. In Udaipur, I was mostly working with reproductive toxicology, and that is when I got an opportunity to visit Cold Spring Harbor, and that was one of the earliest meeting of the human genome sequencing meeting there, which was completely sponsored by this Genentech in those
So
days.
excuse me. Sorry to interrupt you. Are your slides? Are you moving your slides?
No, right now no,
Okay. Yeah. Okay. Yeah. Sorry. Yeah.
but you can hear me, right?
Yeah. Very much. Yeah.
Yeah.
Please, sir. Please.
So in the Genentech, I actually got exposed to human genome sequencing activities. Of course, during those days, it was completely new. Absolutely, whatever they were talking, I was not very clear about it. But I had made some very good friends those days. So one of them went on to become the director of NHGRI called Chandrasekharappa. So Chandrasekharappa used to actually literally help me in learning the nuances of the genetics in those meetings. And then came and of course there had been a lot of discussion that what is going to happen with the genome sequencing, and there was another group which was focusing on the genome diversity activities there. So I could understand that yes, there is a need to address another concern, that is genomic diversity, and that is when where India possibly can play a tremendous role there. But then you have a lot of feedback, but you don't have opportunity to really do the same work back home there. So when I came back, I joined University of Medical Sciences in New Delhi. I worked with Chitra Sarkar, who is presently the president of IAIMS Kalyani, and then Professor Subrata Sinha, who happened to be the director of NBRC. And both of them had been the professor in AIMS pathology and biochemistry there. And from there, we published some extremely good journal publications on oncogene and International Journal of Cancer while working in AIMS itself there. And my mentor went on to publish series of oncogene in those days around 1990s or so. Then from there, I moved to CCMB, and that is when possibly I explored the opportunity of working on the genomics. But then uh, I was not really knowing because this is what you can see. When I moved to CCMB, I worked with Lalji Singh. And you name any animal, possibly have taken a blood sample of all these animals there. Be it lions, tigers, leopards, rhinos, elephants, pandas, lion-tailed macaques. So
Sir,
any human.
sorry, I hope you are now moving your slides, sir. We don't see your slide transition.
So can you see the second slide about the animals?
No.
No?
No.
Can you see the slide? No.
No, no, sir. Unfortunately, no. Yeah, I'm sorry for this. Yeah.
Okay.
Can you stop sharing and restart with full screen? Yeah. You can stop sharing the screen and restart, reshare with full screen. Yeah.
Wait. I have to open the file.
Yes, I have a suggestion. Can you get that PPT from Monik sir and you play from your side?
Yeah, I can do that as a last resort. Yeah.
Yeah. Can you see now?
Yes, sir. Yeah. It's clear. Yeah.
Okay. So just let me move the slides. Can you see the second slide?
Yes, very much. Very much, sir. Yeah.
Okay. Okay. So, and you also have to appreciate in those days, we had no laptops, no the digital computers. So we started from long floppy disk to presently carrying. We are actually carrying this small clouds there. Hmm. So portable clouds there. So things have evolved drastically in a very rapid pace. So I think the people who had been working the last 10 years, they will not never know what had been what we all had come across there. And then in those days, it was mostly about the dermatographics or fingerprints. And from there, fingerprints, we have evolved into DNA fingerprints. And that is when I moved to Kerala, I started working on the forensics also there, and established the DNA forensic lab in Kerala. And then we also went on to work on this what decides the ridges of the dermatoglyphics there. And then of course there are a lot of other parameters like environmental factors like seasons, horoscopes, and all those things. These are all a part of my activities which I keep on doing for various research things. Anyway, this I'll come to it later on. So coming back to this wildlife, so it was a very interesting program that was initiated by Professor Lalji Singh. But unfortunately, while you are working as a postdoc, you're also worried about your career, and that is when I decided to really keep on applying for a faculty position. And during that time, Dr. M.R. Das, who also happened to be the alumni of CCMB. So, and he was a directorate scientist of CCMB. He was trying to start up this institute in Kerala, Rajiv Gandhi Center for Biotechnology. And that is when I came to be one of the founding member of RGCB. And one of the critical component of Dr. M.R. Das was that he lost his Nobel Prize to none other than our yeah, what do you call it? Mm, the person who discovered the RNA, RNA transcription. Uh, Nowadays, I'm forgetting things.
Godwin?
So he was one of the very
See,
pioneering scientists there. Uh,
yeah.
Anyway, so he was And so he lost his Nobel Prize, uh, although he was one of the pioneer in working on discovering the RNA transcriptase assays. Uh, and that is one what has popularized now about RNA quantitative transcription or transcriptive biology. And but then when he came here, he established this institution. He brought in a lot of pioneers in the field. You can see James Watson, then Craig Venter, Charles Cantor, many of these pioneering scientists who had been a frequent visitor to RGCB. And this is the present day RGCB that you can see. We have three campuses, and this is the main campus that you can see here, and this is the new campus that's coming up there. And this is the slide that and when I joined RGCB, my Dr. Imadas asked, what do you want to work on? So I said that I want to work on population genetics, and I want to work with the tribal cell. So he laughed at me and he said that, oh, Kerala doesn't have tribals and all. So then I had a friend here who gave me the list of tribals who exist in Kerala. And this is the 30-year-old slide which I made it to convince him that why it is important to really work on tribal genetics. And so then with that, I moved on to the entire length. And so these are the slides which I really, those days, these are all 30-year-old slides which I made to really convince him that why it is very important there. And this was the booklet that NIH was publishing it. And in this, if you read this book, you'll find out in this particular book, they have specifically recommended that India is a land of genomic diversity, and therefore, if something has to come out, it has to come out only from India then. So this book was also published during that time. And so it also tells you about how to collect samples and how to really proceed with genomic diversity studies and all. And then I moved on to almost all parts of Kerala, right from Wayanad, Kalpetta to Agasthyamala in down south in Trivandrum, and visited all the tribal areas. And by moving these to tribal areas, we collected a lot of blood samples from these. And this is again a 30 years old slide which I made it in my first presentation there. And then we built up the population genetic map of Kerala using first microsatellites and HLA genes. And it was initially done with two of my PhD students. And a part of their work is actually now is written in Malayalam textbooks, and which really created a lot of concern, because they say most often people realize that we are only worried about, we are actually discovering, or we are possibly trying to structure the populations based on the caste and religion and all that. But that was not the intention. Our intention was to really understand the population genetic parameters so that one can really think of understanding what it means in terms of public health point of view there. And then we went on to develop the, I think it got stuck. I think it got stuck now again.
Yeah, but your voice is okay, sir. I don't know why. Yeah, I think maybe the slides are frozen. Maybe you can rejoin, or maybe stop sharing the screen again and rejoin.
Yeah, now you can see, right?
Yeah, it's moving. Yeah, yes.
So and then this was all in 2000 to 2002, when we published the LD map of Kerala populations, again using microsatellite markers of various tribal populations. And of course, this was only about the chromosome number 12 and 13 that we scanned the entire genome of chromosome 12 and 13. And we could find out, yes, wherever you see red and blue, those are the regions which is highly in strong LD between the markers and all. So this is a lot of implications. Whoever are there in the population genetics will realize that what is this implication from the public health point of view. That again it got stuck. Ah. And then we were looking into another gene. Again, this was in 2002 itself, when we did a mutation profiling of a particular gene, which is extensively responsible for acetylation of most important anti-TB drug, which is called isoniazid. And a lot of people had been working on drug resistance TB. But if you see this particular table, you will realize that in India, we are 26% of the Indians are actually rapid acetylators, in contrast to 74% are slow acetylators. But in contrast, the Chinese and Japanese, around 80 to 90% of them are rapid acetylators. So therefore, the drug dosage has to be determined based on the genetic parameters, because if you give a slow acetylator the same dosage, and if you give a rapid acetylator the same dosage, you are bound to see drug resistance in slow acetylator individuals. So therefore, this was then a book someone published where I wrote a foreword for that particular book, um, about the host genetics is very critical in deciding the TB resistance in various individuals. And today we all celebrate this event of genome sequencing in the country. But there had been three people who had been I'll say who had been pioneering these activities. One was Lalji Singh, Samir Brahmachari, and Partha Majumdar, who actually have been spearing these activities. But had they been in the center of the activities, where they had proposed that India should really do the genome sequencing way back in 2004 itself, but no one really took it seriously. But in fact, they were ridiculed to a large extent. But today, India is celebrating for the same thing. So that the disadvantage of doing it 20 years later is, now we are talking about the first phase of genome sequencing there, whereas the Western population is actually talking about the third phase of genome sequencing there. So, which is very critical, that one should not be missing out the bus only because of that. Some people will gain out, some people will take to the prominence. So that is where India could not really be a participant in the International Genome Consortium activities. And then once you get into these genomic activities, once looking at the population genetics, then the issue is that what next? So the next is that this was the front page of a week article that was published way back in 2004, which said that the hopes of the Godson country then. And it said that it had the highest suicide rates, alcoholism, unemployment, crime rates, soaring atrocities, and against women and all. Of course, I will try to deny some of the facts, but still, whenever you see such a front page article, you tend to realize that there has to be something truthful in it. And one of the critical aspect was mental illness there. So mental illness was the most critical aspect, and that is when I decided that I should really start focusing on the brain there. The reason why brain, because if you address the brain, I can address all the sustainable development goals that the world tries to achieve. So therefore, the brain is the center of all the sustainable development goals. And there are several dimensions to brain. I'll not get into all those details. I'll skip many of the slides because of your time constraints, and then build up my theory that what are the main principles of healthcare. The main principle of healthcare is precise diagnosis, precise medication, and can we do disease forecasting. Now let me give you some example of, do we have precise diagnosis then. And then I'll give you an example of precise medication also then. Now, precise diagnosis, if you literally see all the symptoms, many of us possibly might be having these symptoms. During the stress time or during exam time, these symptoms will aggravate. But does this really symptom are a cause of any serious illness? Yes, this can be a cause of serious illness. So based on these symptoms, this lady went to a tertiary care hospital, underwent all CT scans, MRI scans, and all, and then she was diagnosed to have migraine and discharged from the hospital, saying that she was given some anti-migraine medication, and she was discharged from the hospital then. 2 days later, she turns out to be in a completely vegetative state because she had a cerebral stroke which ruptured, and due to which she became completely paralytic then. And it is so severe. If you see this news, in this happened in Jamshedpur, in 72 hours there were 29 patients who died because of cerebral hemorrhage. And the incidence of this particular is almost comparable to breast cancer or AIDS. There, you have you have made a huge issue of AIDS. If you see this particular slide, this slide actually the American Society of Stroke, they try to convince that why it is very important, because the number of people who die due to AIDS is almost 25,000, and which is almost equivalent to the number of people who die due to aneurysms or or cerebral stroke ruptured. But then, so it's very serious, a serious concern, and I'll come to some this aspect again later on. So therefore, the diagnosis can really go upside down. I can give you several examples of the areas that I worked on, where does the diagnosis really fails. Now let us come to the medication. Do we have precise medication? If you see that medical error is considered to be the third largest cause of death globally, and particularly in US. There. So if you can imagine, is the third largest cause of death, and and an average doctor spends close to around 20 to 30 minutes per patient in US. In contrast, in India, the doctor spends only 2 to 3 minutes on an average. This doesn't happen in every. So in spite of spending between 20 to 30 minutes per patient, medical error is considered to be the third largest cause of death in US. And of course, the adverse drug reporting is continuously increasing. If you visit the FDA dashboard, you will find out what is the incidence of adverse drug reactions. And if you read these 4 books, you will really realize the quantum of the issue that I am talking about, and which says that drug discovery is basically needs to be thoroughly looked into, and which really questions a lot of the aspects of the drug discovery process. On an average, 40% of medications don't work very effectively. 91% of drugs that has been established that has not been tested whether it can impact the fetus or not. And many of these symptoms that you see about cleft lip, always ventricular septal defect, atrial septal defects, these all can be caused because of the drugs itself, particularly the anti-epileptic drugs when it is given to pregnant mothers. So in some pregnant mothers, this particular drug can result into those particular abnormalities. And then the paracetamol is known to induce cerebral palsy. It doesn't happen in every individual. It happens only a fraction of the individual. Then. But it's still important that to identify which individual these particular drugs can induce such malformations. Diclofenac can also induce a lot of cardiac-related issues. Then NSAIDs can also induce cardiac arrest. In several NSAIDs can induce cardiac arrest. And of course, then it shows that an analysis shows that on an average, $28 billion per year has been wasted in preclinical research. So therefore, there is a lot of things to be investigated. I'm opening up the avenues for many of you to really take up the research, because my research is almost on the last phases of it. but there is a lot of scope for many of you to really take it forward. And as a result, NIH has initiated a program of double check all biomedical studies. So it's not only in NIH, but even Germany, Brazil, and many other countries have also initiated a similar program on reinvestigating all biomedical studies. So therefore, the diseases that I work on, diagnosis is not straightforward, biology is not very clear, there is no precise biochemical marker, there is no precise physiological marker, no consensus mode of diagnosis. Ethnic and geographical variations are very diverse. Symptomatic variation do affect. Multiple drugs with trial and error approach, and medications can provide a big bazaar offer, buy one get one free. So while you are undergoing treatment for schizophrenia, you can get Parkinson's free of cost. And the disease that I work on is schizophrenia, suicide, epilepsies, and stroke. For the adult conditions, and for the pediatric conditions, I work on non-syndromic deafness, autism, epileptic encephalopathies, and all rare pediatric brain associated conditions. Yeah, so this I'll skip. And what did we do? We actually identified how this particular anti-epileptic drugs, through various epigenetic markers, through proteome markers, and the microRNA markers, we identified that which are the how this particular drugs can induce those malformations and all. So if one is interested, they can take up this particular marker that we have discovered in this particular paper, which was published in Cells very recently, and one can really try to think of validating those particular observations to see whether those particular epigenetic markers or proteome markers or microRNA markers can really hold true for for figuring out some of these malformations that I already shown in one of my earlier slides. That what are the malformations that these anti-epileptic drugs can induce. Similarly, this is another very large study that we have performed, again using antihypertensive drugs. How these antihypertensive drugs, again using proteome-based, this is all proteome, total proteome, and then whole microRNAs and whole methylation markers using 935,000 methylation sites. We have demonstrated that how these particular methylation marks, microRNA marks, can be implicated in various side effects. If you control these side effects, control those microRNAs or protein markers, then you can actually prevent having those side effects from these particular drugs. So this was also recently published that you can see in International Journal of Molecular Sciences. This again I'll skip. This again I'll skip. So so with anti-epileptic drugs I've said, with antihypertensive drugs I've said, now with antipsychotic drugs also we have done again a huge microRNA analysis and the whole proteome, whole methylation analysis, and we have identified several markers which can actually be indicative of the various side effects and all. But interestingly, this same study also demonstrated that the pitfalls of the early publications that people have published with respect to microRNA and marks with respect to schizophrenia. They have reported with respect to pathogenesis there. Now, whatever observation that you see in this particular slide, these are the observations of the drug-induced marks there, which means that the early publications where they have reported about the pathogenesis mark, they are nothing to do with pathogenesis, but they are all drug-induced pathogenesis marks. So this we published an article where we questioned all epigenetic observation that has been reported in schizophrenia. And the reason why we questioned is because they were all done on patients who were already on medications or postmortem samples, which means that they have nothing to do with pathogenesis marks. They are all drug-induced marks. And this we have demonstrated using this particular data. So as a result, we say that if you want to do the complex genetics, you have to address these following facts. One is phenotypic diversity. You have to address the epigenetic diversity that exists. You have to have the genetic diversity that exists and the comorbid factors that associate with conditions. So if you don't address these issues, you are bound to actually fail in very classically demonstrating that what is the real cause of the disease. So I'll stop here. In case if you want to have actually observations for each and every component separately, but I think I'll stop it here and I'll take up any questions if anyone has it. And or if you want to see more on this aspect, I can show you on subsequent when you raise the concerns and all. Right?
Yeah,
Yeah,
thank you so
thank you,
much.
sir.
Thank
Thank
you
you
so
so
much,
much,
for that
for that
illustrious
illustrious presentation
presentation
and
and
very
very
detailed
detailed
and
very
very
nice
nice
presentation.
presentation.
So,
So,
first
first
we'll
we'll
go
go
for a
for a
few
few
questions,
questions, sir,
from my
from
side,
my
and then
side, and
we'll
then we'll
request the
request the
audience if they
audience
have
if they
some
have some
questions.
questions.
Um,
Um,
Sir,
Sir,
one of the
one of the
major
major
avenues
avenues
that
that
you
you
have
have
really
really
worked was
worked
on
was on
multi-drug
multi-drug
resistance.
resistance.
So, if
So,
you really
if you
look
really look
into
into
the
the
kind of
kind of
MDR
MDR
associated
associated
pathways
pathways
that
that are
inflicting
inflicting
certain
certain
diseased
diseased
phenotypes,
phenotypes,
what amount
what
of
amount of
diet
diet
parameters are
parameters are
crucial
crucial
for such
for such
epigenetic
epigenetic
studies
studies
associated with
associated with
MDR, sir?
MDR,
I
sir?
mean, do you
I mean, do you
see
see
any
any do you see
just because
just
there is
because
a
there is
diet
a
as a
diet as a
factor, or
factor
whether there is a
or there is
diet
a
as a
diet as a
cofactor,
cofactor,
there is a
there
more
is a more
emphasis on
emphasis on
MDR
MDR
or
or
No,
MDR
no.
MDR
associated
is
What specific factors?
diet,
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 Diet.
diet.
yeah,
Yeah,
Yeah, definitely diet is one of the major component. So in fact, that would have come in one of my epigenetic diversity there. Since you raised this, uh, what diet you are actually taking it can also be determined based on your genetic parameters.
right,
right. Yeah,
So,
right,
so therefore, I'll say that uh, all this phenotypic diversity, epigenetic diversity, that can all be addressed using genetic diversity.
right,
I
right,
did an editorial on this particular aspect that although we simply brush away all the diseases like complex diseases like diabetes, hypertension, stroke to be associated with 50% genetics and 50% epigenetics. So I
right,
say that
right,
it is not true. The 50% epigenetics that you brush away is also being determined based on your genetic parameters.
right,
right,
right.
right. Yeah.
Wonderful, sir.
Wonderful, sir.
So
So
moving
moving
further,
further,
in one
in
of
one of
your
your
particular
particular
slides,
slides,
there is a
there is
high
a high
linkage
linkage
disequilibrium.
disequilibrium.
That is
That
what you
is
have,
what you have,
Right.
you have
you have
shown.
shown.
So if you really
So if you really
see,
see,
that has
that has
also been a kind
also
of
been a kind of a
major
major
challenge. Say if
challenge.
the
Say if the
SNPs are in
SNPs are
high
in high
LD,
LD,
identifying
identifying
a
a
causal
causal
variant to
variant
that
to that
particular
particular
disease
disease
phenotype is
phenotype
really
is really
challenging.
challenging.
So
So
since you have
since you've
worked
worked
on
on
aneurysmal
aneurysmal
diseases, so
diseases, or
neuronal
neuronal
dysfunction,
dysfunction,
including
including
schizophrenia,
schizophrenia,
probably
probably
dementia,
dementia,
Parkinson's,
Parkinson's,
Alzheimer's, or
Alzheimer's,
any
or any
neurological
neurological
diseases,
diseases, assuming
assuming
that
that there are
there
certain
are certain
SNPs in
SNPs
high
in
LD,
high LD,
what
what
amount of
amount of
variant
variant
annotation
annotation
attributed
attributed
to
to
your
your
particular
particular
study,
studies?
sir? How did you
How did you
prioritize
prioritize
those particular
those particular
SNPs in high
SNPs in
LD?
high LD?
No, no. So that LD mapping was done using STR based mapping, microsatellite based mapping.
That is
Oh,
not
that is
based on
not based on
NGS.
NGS. Okay, yeah,
No.
okay,
Okay,
So
okay, yeah,
that was almost 25 years back.
right,
Uh,
right,
During that time, SNP mapping was still not very popular.
right,
right.
So
right.
Right.
Wonderful, sir.
Wonderful, sir.
And moving
And moving
further, I think
further,
you
I think
raised
you
a very
raised a very
important point
important
about
point
those
about those
three
three
big
big
persona
persona
about
about thinking of
thinking
starting a
of
human genome project
way back
way
some
back some
25 years
5 years
ago.
ago.
That would have
That
been
would
a
have been a
crucial
crucial
starter. In
starter. In
fact,
fact,
we
today
would have
we would have
probably
probably
gotten
gotten a
mammalian data,
mammalian data
because
because
I
I
also
also
akin that
that particular
knowledge to
knowledge
the
to the
lack of
lack of
democratization of
democratization of
genome India
genome
data
India data
even till
even till
date,
date,
to be very
to be very
honest.
honest.
And
And that
that
also
also
lacks some
lacks
kind
some
of
kind of
a
a
gold
gold
standard
standard, or
or a
a
lack of
lack of
reference
reference
data,
data,
reference mapping, with respect to Indian population as well. So, so where do you see, sir, maybe the next 5, 6 years as more and more Genome India projects are coming up? Where do you see the democratization of these Genome India data coming up? What is
Yeah,
your
so
purview on
you
this?
literally asked me this particular question. I'll say that India was not far behind. India actually was started its own genomic activities long back. Like if you see the one which I have presented, I have also started almost way back in 25 years back. But then I'm not a part of the Genome India program there. So,
Right.
but the issue, and similarly, whatever Lalji Singh, Samar Brahmachari, and they had already initiated that particular program, but many of this data is not available in the public domain. Then. So we have to rediscover again and again every time we approve such projects and all. Unless this particular data is available in the public domain, nothing. So I literally wrote an article on Economic Times a couple of years back saying that how many people have actually developed these database portals in the respective publications, but none of these databases are actually available in the public domain. In India itself, there are more than 12 to 15 such databases where different people have claimed to say that they have actually developed a portal there. But none of these portals are actually available there. I have actually made up a map also. Very soon you will see those same maps in my blogs coming up that which says that yes, there are India had been in the forefront, but many of this data is not available in the public domain. We will keep
Right.
on
Wonderful.
time there.
Yeah. Yeah, yeah. Thank you, sir, for that. And you have a very wonderful dual distinction of working for, of course, population genomic data and as well as the clinical genomic data. But if I'm not digging too much, do you think that you were a bit fortunate that probably you were in Kerala and the health sector is probably good? That could have probably given you, a good amount of, genomic what you call data coming from various phenotypes as opposed to probably other states where the health is not considered that kind of a major entity. Yeah.
Yeah, so normally what happens is it always goes with the clinicians there. So we came across some very good clinicians who actually were
Right.
quite aggressive in nature there.
Right.
So as a result, we could actually achieve many of this work there. But say similar clinicians are there all over the country there. So I can tell you one of my friend, Durpan Bharadwaj, he actually collected close to 80,000 samples of diabetes.
Right.
So which is a huge sample size there. So
True.
similarly, I think there are clinicians in the country. Yes, Kerala has one of the advantages that yes, there are people who are very passionate about their health. In North India, even if you have a small health concern, they will try to manage by themselves there. They will not go to the hospital and all. In Kerala, that's not true. They will actually, for each and every small thing, they will go to the hospital, they will consult the doctor, and that is very, 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 very useful for many of us.
Right.
So that
And
awareness
not only that.
is very good there, but at the same time, over awareness is also very dangerous there. So when we
Mhm.
publish this particular data on articles on the population genetics, I had received calls from so many places there, and but then some were very threatening in nature also. But this wouldn't have been in this scenario in any part of the country there.
Right. Yeah. Yeah. And of course, moving back to your population genetic studies, that way back some 30 years when you started, when you initiated the tribal genomics data, Kerala tribes and probably to a certain extent South Tamil Nadu tribes, they have a very distinct, phenotypic driven pathways. Probably you don't find those many Kerala tribes associated with probably hemoglobinopathies, or maybe they're not probably that sticklers when compared to maybe Andhra tribes or maybe Maharashtrian tribes or Madhya Pradesh tribes. So, do you see something related to the diet? Is it because there's a large amount of Piscarian diet, a large amount of fish-related diet that is largely associated with the southwest coast of India? Yeah, thank you.
Yeah. So you are repeatedly coming to the same scenario. I'll tell you the same about about the phenotypic diversity. So these are a set of symptoms that associate with schizophrenia, and what you can see is unusual thought content and uncooperativeness is very critical of is very remarkable of Kerala patient population, the schizophrenia patient population there. But this was not seen in the Chennai patient population there, Chennai schizophrenia patient population there. Now you may ask is that if I literally have to pool this set of samples of Kerala and Tamil Chennai, I can actually do blunders there. But then when I try to segregate this patient population, I can say that yes, there is a specific marker that associated with uncooperativeness and a specific marker that is associated with unusual thought content, and that is very typical of Kerala patient population, which you don't try to pick it out in the Chennai patient population. So that is
Right.
where so this was the first time where people started talking about the symptomatology of schizophrenia. Genetics of, so when we start talking about symptomatology of schizophrenia, during that time no one even thought of it. But now many people are trying to talk about the symptomatology of schizophrenia using genetic parameters.
Right. Wonderful, sir. Yeah, that's a great point. Thank you so much. Last couple of questions. Since I also work on pediatric congenital anomalies, I mean, I was a bit intrigued looking into your syndromic anomalies, syndromic, pediatric cases that you have worked on. How distinct is the correlation, or how distinct is the genetic variation that is attributed to, say, syndromic congenital anomalies as opposed to the non-syndromic? I mean, getting a non-syndromic data largely associated with child diseases is not really, I would say, clear, or not really obtained, in India when compared to the syndromic. I mean, because we really don't accord or honor that kind of electronic health records. So, did you face any kind of challenges in these
Yeah.
directions?
So when it comes to syndromic and non-syndromic, the one which is very well classified is with respect to hearing loss. So we have worked on the hearing loss on non-syndromic hearing loss. So and the common features of non-syndromic and syndromic hearing loss is basically the immune genetic parameters. So the syndromic genes also are influenced by the immune genetic parameters, and the non-syndromic ones also are initiated by the immunogenetic parameters. So therefore, I will say that there is very thin line of distinction between syndromic and non-syndromic there. Only the triggers are the one which differs there.
Right, right. Wonderful, sir. Yeah, thank you. Thank you so much. I just have one or two questions, but I'll reserve it later. But Jayaraman sir raised his hand. Jayaraman sir, would you like to ask a question to
Hi, sir.
Moinakl,
Good
please?
evening, Professor Moinakl.
Good evening, Dr. Jayaraman. How are
Yeah,
you?
I'm fine, sir. Yeah. So I have a very general question to ask. You have shown this many diversities along with comorbidities factors coming in. Is precision medicine possible at all? And if it is possible, how do we consider multiple interactions between these? Yeah.
I don't know whether my system, the problem is there with my system. I'm not able to hear very clearly your voice there.
Yeah, I can comment on that, sir. So what sir was asking is, amid the plethora of diseases that you have worked, how relevant is precision medicine, in the diseases of interest that you have worked on over the last several years?
Precision medicine, yes, it can help a lot. So only thing is you have to identify the markers for it, and we also have to identify what is the drugs that has been given there. So since in the clinical scenario it's very difficult to get a very precise clinical data or the drug data, so therefore we have to really try to work out from the in vitro data and then try to validate how many of this particular genetic or pharmacogenetic or pharmacoepigenetic marks are applicable in the patient scenarios there. So which we have published a couple of years back about pharmacoepigenetic marks that we relate with respect to antipsychotic drugs, and those particular epigenetic marks how they can be really taken care of it. That I think that we have not worked on it, but one can really start thinking of working on it.
Yeah. Thank you, sir. Jaraman, sir, thank you. I
Thank you.
hope that answered your question. Yeah, Prajwal, please unmute your microphone. Yeah, and ask. Yeah, Prajwal. Prajwal, you raised your hand. Can you please unmute your microphone and ask a question? Yeah. Yeah, Nirmalo, yes, sir, please. Yeah.
As usual, it is pleasure listening to Moinakl, and since I have been associated with him for quite many years, and just wanted to know that one of my students, Dr. Paneer Das, I don't know whether he was working at the time when you were there at CCMB. So he worked with Dr. Lalji Singh and also P.D. Gupta. He was from Jaipur. He was there for a number of years in CCMB. And Dr. Shivaji, my friend, he was there. So what my question is that probably they were working on the endangered wildlife species. And is, is there any information about what is the current scenario about the National Institute of DNA fingerprinting and diagnostics, which was established later, only focusing primarily on the endangered wildlife species?
Yeah, actually, after me leaving that particular activity, CCMB actually came up with another center called LACONS. So, Laboratory for Conservation of Endangered Animals. That's a phenomenal institution there. And so now a lot of the DNA samples, we also collect sperm samples there. And both the DNA samples and the sperm samples, I was involved in visiting all the places and collecting the DNA blood samples and the sperm samples there. So we made a repository of many of these sperms and blood samples in the LACONS there. So which is still available there, but not much of work has been carried out in those particular samples. I don't know why. I'm also very intrigued why people are not taking it forward there. So we must have collected more than 200 lions, and the story of lions. So how the entire project started, because there was some rumor that all the lions that are actually available in the Indian market, they are all hybrid lions. The moment anyone labels it as a hybrid lion, you know that its fertility rate is going to be drastically less there. So through our genetic studies, we convinced that none of the lions actually are hybrid, and they don't even have, and their fertility count is absolutely fine there. So their sperm count is actually quite motile there. So that rumor was completely taken off there.
Yeah.
Okay, thank you so much.
Yeah, wonderful, sir. Any other questions, please? Otherwise, I just have last two questions for sir amid his busy schedules. So one of the other questions that I wanted to ask you was, there is a very typical end disease phenotype called SUDEP, sudden death in epilepsy. Which is SUDEP, sudden death in epilepsy. Yeah, which is not really that drastically studied in India, albeit the fact that if you see, South Indians have a large amount of sudden death in epilepsy, attributing to almost 56% of the total Indian population, as per the prevalence that is known. So is there any specific reason why such particular diseases are not studied? I mean, is it because of lack of sample acquisition, or where do we stand up front on that?
So the biggest challenge that comes is actually about the clinician's commitment towards the study. So if there is a strong commitment, like now I've shown you about stroke. When I submitted this project way back in 2002, they said, no, this is not a important program for the country, and it was rejected. But then now you know that how important it is. So unless it happens to someone close to the funding bodies or someone close to the politicians, nothing is going to happen seriously. So possibly I would say that one has to really start convincing it how critical or how important this particular condition is through various evidences, and start writing articles on those particular aspects.
Right. Thank you, sir. So one final question which I usually ask my Sciversation guests is, are there any occasions where you have disagreed in an agreeable way or agreed to disagree with any of your colleagues, be it your seniors or your students or any other things? Then how did you put that disagreement in a more agreeable way so that, the younger generation here can learn from such kind of things? Yeah. Thank you so much.
So, in fact, I don't really debate any disagreement, because disagreement is actually a very critical component of one's learning process. And if someone disagrees, it means that he or she has an opinion there, which is very important there. So if I say that whatever I am doing is the best thing, then that actually curtails your growth. So I would really, many of my students actually disagree with me, and I really appreciate that. And I think that's good for their growth as well as my growth, and also my understanding, and also my investigations.
Wonderful, wonderful, sir. So that's a great point, friends. That was Banerjee. So sir needs no mention. He has put up his entire 3.5 years, more than 3.5 years of decade of experience today in his Sciversation. In fact, if really look into several of his publications and also certain opinion articles that he has written in various leading dailies, that shows his large amount of love for science, and looking at his magnanimity, there is nothing of any of that particular sort that curiosity has got its own reasoning. And if you can look into his wonderful LinkedIn profile, he gives a wonderful narration on philosophy as well, on wonderful way of putting the philosophy, and the final response speaks volumes about his about his nature and his scientific excellence. So with this last question, I think we should give a big round of applause to Banerjee, sir. Thank you so much, sir, for your wonderful Sciversation. Can I request everyone to switch on your camera, please? And can you stop sharing the screen, sir, so that we'll have a group photograph? Yeah, thank you. Yeah. So,
if anyone is interested, they can write to me. I am always available there. Oh, Rashmi, I can see.
Yeah, Rashmi is here. Rashmi is always there in our meetings, sir. She has been, she would always attend all the meetings, and thank you so much. Yeah. And 1 minute, I'm just trying to, yeah. Raj, can you click on your camera? Sovan? Velu? Devesh Dorang, please. I can't make an AI photograph of yours. I'm sorry. So I have to take a good photograph. Yes. Okay. Say cheese. Yeah. Thank you very much. Thank you so much, sir. Thank you so much, Moinakl, for your time. And please, let's give a round of applause.
Thank you very much.
Thank you, sir. Dr. Moinakl.
Thank you, sir. Nice to listen to you.
Okay, bye. Thank you, Jayaraman.
Thank you.